Hello everyone, welcome to my channel, the Biology Club. So, we were learning about the topic Animal Kingdom. So, we have done two parts of this particular topic. So, will start with the part 3. So, let me tell you what we have learned before. Let me brief you about it. So, we have learned about the level of organizations, the symmetry, the body cavity, which is also known as the coelom, the and the different types of phylum. Right. Now, as we all know, Animalia kingdom consists of multicellular organisms, right? Now, in multicellular organism, we have first level of organization, that is cellular level of organization. Okay, and we have other level of organization, that is tissue, organ, and organ system level of organization. Now, if we are talking about the level of organization which is cellular, we all know it will be having a symmetrical symmetry, and it will not process any coelom in it. So the phylum will be Porifera here. Likewise, we have different type of level of organisms which have different phylums example. So let's start with the phylum Porifera. So the phylum Porifera, commonly known as sponges or the pore bearers. Now, why they are known as pore bearers? Because in the sponges, you can see there will be many minute or tiny pores, right? That's why it is named as pore bearers also. Pore bearers, the thing which will have lots and lots of pores. That's it. So we can see one of the example here. So you can see there are so many pores present in this particular sponge here, right? Okay, let's start with the phylum Porifera's general features here. Now, you can see here, we all know that Porifera's habitat will be aquatic always. It is aquatic every time. Okay, it can be freshwater or marine water. Now, if it is freshwater, the common example will be Spongilla. I will show you the diagram here. So, this is one of the very common example, that is Spongilla. Now, uh, after seeing this spongilla, let me tell you one very important thing about habitat. Now, sponges are sessile organism, which means they will attach to some object, right? Now, suppose this is our sponge and it is attached to this particular rock. Okay, this is attached to this particular rock. Now, this particular rock will act as substratum for the sponge here. Okay. Now, how will they obtain their food? They will obtain their food through water entering their body. Okay, the water will enter in their body, it will bring food with them, and it will come inside the uh, body of the sponges. Okay, now, as we all know, the body symmetry is asymmetrical here, right? Body symmetry is asymmetrical here. Also, if we will talk about level of organization, it is tissue level of organization or cell level of organization, cellular level of organization. Which one? Of course, it is cellular level of organization. Just now we have seen it, right? It is cellular level of organization. Now here, what will happen in this organization? All the cells will come independently, and they will form, they will perform some kind of function, but they will not form the tissue. They will coordinate with each other, they will help each other, but they will not form any tissue. Means this cell, this cell, this cell, this cell, this cell, this cell, all are independent. Okay, but for some task, they will form a group and do that task, but they will not form any tissue. This is important here in cellular level of organization. Please remember, the level of organization here is cellular. Body is asymmetric here. Fine. Okay. Okay. Now, let's talk about the body form here. Now, body form is again very, very, very important. Okay, let me tell you here. Suppose this is the sponge we are talking about. So it has many minute pores around it. So this minute pore are known as ostia here. Ostia. This minute pore is known as ostia. Now, inside this, if I will take the longitudinal section of the sponge, you will see one cavity inside it. Let me draw that cavity with this color. If I will take a longitudinal section here, you can find one cavity here. Now, this cavity is known as spongocyst. Spongocyst. Okay, this cavity is known as spongocyst. Now, what will happen? Whatever things are coming inside through the ostia, because through pores only water will enter, right? Okay, it will come inside this particular cavity known as spongocyst, and also when it will uh, means from the spongocyst only, the water exists through a large opening called osculum. So here there will be one opening. Okay, so exit of water through this opening, okay, will happen when it will not need that particular kind of water, because it can be excreta of that particular organism, right? So this opening is known as osculum. Okay, so the unwanted thing will go out from this particular opening. Now water enters through this ostia, brings the food, oxygen inside the body of the sponge also. Please remember that. Now let's talk about this particular system here. Fine. Let's talk about this particular system here. This system is known as canal system. The entering of water through this particular channel is known as channel system. So the body of sponges, okay, the body of this sponge is organized in such a manner as to form a complex system of pores called canals. So the structure I have shown you just now, okay, it is known as canal system here. Okay, it helps in food gathering, respiratory exchange, removal of waste, and transfer of gametes also. Okay, so let's see how many type of canal system are present here. There are three types of depending on the sponge's shape of the sponge, there are three different types of canal system present here. Ascon type, Ascon type you can see in leucosolina. In Sycon type you can see in Sycon. Let me write out the example. Leucosolenia. Okay, Sycon, and for Leucon type it is Spongilla. Fine. Okay, now let me show you each and every type of canal system. Okay, so for Ascon type you can see this is how the Ascon type will look like. Okay, there are certain gaps. Now these gaps are nothing but the tiny pores present here. Okay, and water go out through this particular opening known as osculum. Then we have Sycon type. Okay, we have Sycon type. So this is how the Sycon type will look like. It is like this. So they are like wavy like this. Okay, and other things are similar. Just the outer structure is little bit different. Then we have the Leucon type. Let me rub this first. Now this is how the Leucon type will be. Okay, this is how the Leucon type will be. I hope it is clear. As you can see here, so here the distance of between the spongocyst and the outer layer is much more than the other type of canal system in sponges. Okay, now let me talk about the digestion. It is intracellular, and the skeleton of this particular phylum are you know almost they have the internal skeleton called endoskeleton. Okay, now this internal skeleton provides the support to the body, and sponges are comprises of very small fiber like structure known as spicules. Okay, let me write it down. They will provide support to internal body. Internal body. Okay, so because they're present inside, right? Now this is how it will look like. Let me rub it. Okay, as you can see, this is the inner structure, or you can say endoskeleton of the phylum Porifera, right? So you can see there are porosite, water flow is there through this tiny pore, spongocyst is there. Now in the spongocyst there are many carnocyte or spicules. So spicules or the spongin fibers are present there. Okay, here. So this consists, or this will make the internal skeleton of the sponges. Okay. Now let's talk about the reproduction. So all sponges are hermaphrodite, means sexes are not separated here. Okay, now type of reproduction in sponges. Some will do asexual reproduction like fragmentation, and some will do sexual reproduction, means exchange of gametes here. Development in the sponges, in the development thing, the adult sponge form diploid zygote in the indirect, which means it is mediated by an intermediate free swimming flagellated larva stage for dispersion of species. I will show you how it will happen. That means the larva stage, you can see how it is morphologically different from adult. Adult means morphologically different means, or distinct means physically they are different. Let me show you the diagram here. So you can see here they are fertilized zygote here, right? They will process meiosis. Now after the blastula stage, you can see the hollow sphere. It will form something like this. Then they will develop into larva. See, you can see this is the larva stage. Okay, now slowly they will go and settle down to some substratum. This is a substratum here. Okay, then development collar cell will be there. Slowly it will the collar cells will develop here, right? So this is the development of Porifera. Fine. Now there are some different examples here. Leucosolinia, it is look like this. Euplexitella, it is looking like feather, right? Then Spongilla we have seen just now. Euspangia, uh, Euspongia, it is something like this. Okay, Hyalonema is something like fan. Okay, fly. We will their hand fan is there, right? Just something like this. Then we have Saigon. So we have different type of example in your book. Also, you will find different type of example. So that's all for today. I want to tell you one thing. Please focus on the NCERT, because you will find these all things inside your NCERT only. I have not referred any other book rather than NCERT to teach you also. Now, when we will go to question answer session, obviously I have to take some help rather than NCERT to other books. But if you will understand NCERT, this all things will be very easy for you. Okay, that's all for today. We will meet in our next video. Thank you.